it's a big room, and there is I'm not saying that there is a sort of homeopathic dilution going on, but don't be afraid to come to the forefront. None of the speakers bites, and so maybe a little bit more kosher. My name is Daan Crommelin. I'm chairing this session, and I'm supposed to do the time management. That means that every speaker has 12 minutes, and then maybe 1 or 2 to finish up. Then there is hopefully time for just questions for clarification purposes, and then we move on to the next one. At the end of the session, we ask the speakers to sit here behind the table and answer questions, or maybe ask questions to other speakers why the hell they were saying this or that. For those of you who just choose at random a room, this is the room where we talk about non-biological complex drugs. And it's here on the screen already for a while, so hopefully you have taken that up. The session is organized by a working group, and the working group is called the NBCD working group, the Non-Biological Complex Drugs working group. What are non-biological complex drugs? They are not a biological, so biologicals are not on the table today, and they are not homomolecular structures, so in other words, they are diverse structures. They cannot be isolated and fully quantitated, characterized and or described by physical, chemical, analytical means. That's a very important characteristic of this group of drug products. And last but not least, their product characteristics depend heavily on the manufacture process. So if you have read this, if you have understood this, and you still want to be with us, you're welcome, and otherwise you can go to some other room. Well, for those of you who say this is I'm suffering from a postprandial dip, then everything that's being said today can also be found in this book that just came out. This is the paperback copy, so this is just a pre-print. Just came out, and everything you always wanted to know about MBCDs can be found there. Without further ado, I would like to invite the first speaker, Scott McNeil, Director of Nanotechnology Characterization Laboratory, to give his presentation with the very, very interesting title, Overview.
Very good. Thanks, Daan. So part of the tasking to me was to give an overview, but also I took the liberty of saying, well, if I get to give an overview, I get to also set the stage and stir things up. So knowing my mischievous nature, I'm going to give you a slight overview and lots of stirring up. So here we go. NCL, or either myself or our staff, have presented, I think, at least 3 times here at CLINAM. So I won't belabor the point of what we do, just simply stating that we conduct preclinical characterization of nanomaterials, which, if I got the ontology right, is a subset of NBCDs, nanomedicine, that is. We've characterized over 300 materials. We have 10 of those that we've generated data for or in clinical trials. So we see strengths and limitations of each subset and each type, and we see trends in biocompatibility in what works, what doesn't, and we're, to perhaps state the obvious, we're very fluent on characterization. And as Daan just said, the physical chemical traits of these NBCDs do not define the material, as hopefully you'll see here in just a few minutes. Okay, so we hear a lot of hype about how a nanotech formulation can add benefit to an existing formulation. So I use this as just now a legacy case study of something we saw perhaps over 8 years ago. This is a gold particle that delivers tumor necrosis factor alpha. TNF alpha was used in the late 90s in clinical trials, but it failed miserably because it actually killed some of the patients due to immunotoxicity. By attaching that same drug to a gold nanoparticle, we can greatly and dramatically change the PK properties. So here is, in red, is free tumor necrosis factor. It has a half-life, as you see, of about 26 minutes. Attaching that particle, or that same drug, to a gold particle increases that to 182 minutes, almost as much as the free gold, the carrier. So this is an example, if my slides work right, there it is, of how just simply attaching the same API, the same drug, to a nanoparticle can dramatically change the PK properties. You'll have to take my word for it that this also holds true for decreased toxicity. So we can engineer properties into and out of just using nanoparticles, the field that we're all in. Okay, so how is it different, though, than the legacy drugs and biologic? I mean, if you're a reviewer, for example, you have two buckets that you can put drugs and formulations into. You can put it into the small molecule, or you can put it into a biologic. But as you'll see from some of these case studies, the nanomedicines, the NBCDs, fall in between the two. They have properties of each. They're very complex with heterogeneity. They have some characteristics of small molecules. Often they carry a small molecule, as in the case of Doxil. And they have some characteristics of biologics in that the nanocarrier itself can function as part of the mechanism of action and or be functionalized with a biologic. So simple stuff, right? So just to recap this a little bit, I don't have enough time to give this true due diligence, but if you submit a package, an IND to the FDA, or sorry, it's actually if you're presenting your formulation as a generic, a follow-on, traditionally with a small molecule, the parameters that are heavily weighted are PCC, physical chemical characterization, the size, the molecular weight, and so on. So that's fairly easy or fairly discrete to define. On occasion, there may be a need for bioequivalence or others as deemed appropriate. So we agree with that, certainly, that each case is unique, each submission is unique. And for the case of biologics, it's far more rigorous in that you may actually have to do an efficacy study. Not to belabor the point, but just as a rule of thumb, efficacy studies are more often required in the EU than the US, but certainly you have to show bioequivalence if you're declaring the concept as a biosimilar. Okay, but what about nanomedicines? Well, as I've been saying, they fall right in between. Well, if you have something like Doxil, yes, you can do drug release kinetics, you can do PK, and more or less it behaves like a small molecule on this side. But other concepts, as the one you'll hear later on from Neil Desai, Abraxane, it falls in between because, yes, it carries a drug, but there's some evidence that the carrier itself contributes to the mode of action. In this case, the albumin assists with the transcytosis of the drug and then contributes to efficacy. So how do you compare a follow-on if you have that mechanism of action? Well, hopefully you're not just going by the PCC, because you'll never get there as far as the data package necessary to compare the follow-on. And so here's an example with Lipidox that is now approved. Initially, when Lipidox was introduced in the US, it was done so under executive order because of the shortage of Doxil. It is now fully approved by FDA, but not yet, to my knowledge, by EMA. But you'll see there the difference between those two is that you have to identify what parameters, what measure end you need to track in order to do that bioequivalence study. In this case, it was, well, I'll get into this in just a second, but it did require a PK study. and a PK of the drug itself, not just the total drug. And what's the difference between those two? Stay with me, it's going to get fun. All right, so there's more nanomedicines that are coming as follow-ons. These are now, this information is now public available, but companies that we interact with, one is Azaya, which is another version of Doxil. It's a generic version of Doxil. And Sorrento Therapeutics, which has, it's now in clinical trials as a follow-on to Abraxane, and they're actively involved in their clinical trials. So it's important that we get it right as far as how to characterize these materials to make sure that we're not missing that important measurand that may make or break this concept, because the alternative is that, yes, it may pass a safety requirement, but then if there's a crossover study, yes, they met that criteria, but then the actual clinical study to compare efficacy is done in humans in a non-clinical environment. Or a non-clinical trial environment. It's done on a commercialized product. So then the determination of whether or not it's working or not comes to the clinician in the field. And as discussed with some of you, collating that data may be a little bit difficult to do, where a physician, a clinician, an oncologist, after months or perhaps year, may have a population set enough where he can say, well, I'm not seeing the same equivalency between these two drugs. So you can see how complicated it could be. But a better solution is if somehow we can identify the differences and what that critical parameter is. All right, so here's an example of all the various parameters that we characterize and that we're heavily pushing as far as including in the characterization of the follow-ons. Some terminology here is if you have a nanoformulation, you can have encapsulated, you can have bound, you can have unbound, you can have unencapsulated, and you can have free drug. Got that? Well, what does that mean? Well, the total drug is everything that you would see, whether it's bound or unbound. And because these nanomaterials often have, as I showed you, different release profiles, it includes the encapsulated drug, the drug that's bound in the particle, it includes the unencapsulated, it includes the just free drug, and it includes the protein-bound drug. I'll give you an example of this here in a second. So which one do you characterize? Well, we characterize all of them, but it is very important because the total drug profile, as it says there, is dominated by this. However, this species right here is the species that is free to interact with its target and also is the species that causes the toxicity. So then how do you study bioequivalence? Well, the way that we do that is by evaluating the total unencapsulated and encapsulated drug fractions, not simply the total drug fraction. And getting the assays to do that is can be very laborious because it's very easy and traditional to characterize the total drug and then maybe use a chromatography method to attempt to identify the unencapsulated, but then there's subspecies of that. So here's what I mean by encapsulated, unencapsulated. So there's actually a kinetic, a reverse kinetic between the encapsulated drug in the nanoparticle and the free drug, and that free drug's lifetime before it's bound by proteins in the blood, specifically, well, most likely albumin. Under that, so here's encapsulated and here's unencapsulated. We absolutely need to evaluate that drug release profile. That can be exceptionally difficult to do, but it is germane to be able to do this so that you can truly monitor bioequivalence, and I'll show you why here. So, and why it's important, and how it can actually be predictive of some of the results we'll see in vivo. Not to belabor the point, but we've presented this before in that there's a difference between approval of liposome, the generic liposome, the follow-on, compared to Doxil, and Calyx, which is approved in the U.S. And there's also two different lots of Doxil. the previous law that was discontinued due to process and the one that is currently made. Okay, so just very quickly, if we compare doxil versus liposomal doxil, the follow-on, we get pretty equivalent PK profiles. However, if we compare that same drug with Calyx, the PK profile is different. And because of this, that drug, the follow-on Lipidox, is not approved in the EU. Okay, so why though? Why did they see that difference if it's the same drug, more or less? Well, it has to do with how they analyzed the material as well as the power of the sample size that they use in the clinical trial. Okay, so back to what I was saying about the various methods. Many of these are legacy methods, but they're designed for small molecules. And as I've said now 10 times, nanomaterials, NBCDs, do not necessarily behave as small molecules, especially in identifying these various species. They each has inherent flaws, inherent limitations, and strengths. So what we've been doing recently is developing a series of new assays, one in particular that uses a stable isotope to be able to monitor the various species. Here's some more data on that Sun Pharma, which is the Lipidox example, compared to Doxil. So what happened is the number of patients was far too small for the Calyx study, which resulted in a denial of achieving bioequivalence. You can go further into that as to why there was a difference between that. So the power analysis was correct in addressing the variability of the total drug, but in what we found is that it's actually a measurement of drug concentrations in the various species that you can follow the variability in the clinical study if you follow the variability in the free drug. Let me say that again. If you follow the total drug, you're going to miss it, and your power analysis will not be high enough because there's too much variability. But what we saw is the variability in the free drug nicely mapped to the clinical study. Okay, Dan's over here yelling at me, so I'm going to wrap it up fairly quickly. Some of the characteristics that we recommend as germane to the follow-on package are obviously the PCC, but we often have to generate new biological methods to go after that free portion. And to evaluate both the total drug and the unencapsulated, which is the free drug, and being able to validate that system to know that it is a free drug. In summary, hopefully I've said it and you'll hear it several times, that nanosimilars or NBCDs are not the same as generics or biosimilars, and it requires assessing the various drug fractions, and you may need new bioanalytical techniques. However, it's kind of interesting, and maybe I'm going out on a limb to say that if you can get this right, they may actually be predictive and predictive under in vitro conditions. We've seen that with some of our other assays, so that would be very nice, especially if we can begin to know a priori how that clinical trial is going to come out. Again, that's a bold statement. Okay, so the smart people in NCL are Steve Stern, our toxicologist who's developing these bioassays. All of our immunotox is done by Marina Dobrovolskaya, and Jeff Clogston is now heading up physical-chemical characterization because the FDA stole my deputy Anil Patri, not that I'm resentful about that. Okay, thanks for your time.
Thanks, Scott, for an overview, pointing out what sort of critical elements are out there. Questions for clarification purposes only. Otherwise, we will move on and we'll come to the question and answer session later on. Okay, Neil?
So your last slide was that if you can sort out the free and bound drug, then maybe you're sort of in a good shape to predict bioequivalence. you know, a lot of times the free drug is the smallest fraction of the total drug. Inherently in those methods, you're running into larger errors than when you have, you know, say measuring a bound drug. Now, so how does that play into all of this? You know, you're looking for very small amounts, inherently larger areas, errors. There's additional methods that are required to separate free drug that can lead to additional errors. So how do you sort all that out?
Yeah, well said, and a great question. So what we do is we attempt to optimize it and validate it and learn the level of detection, the level of sensitivity. You need a appropriate method to go after that, whether that's, you know, legacy LS mass spec or more sensitive methods. But again, we try and optimize it in vitro before we go with the clinical samples. And then do the calculations to make sure that the level of sensitivity that we see in vitro is actually going to map over, because as you said, if there's that much variability, then it's exceptionally difficult to make any conclusions. But other than agreeing, there's not an easy answer to it other than the in vitro validation. Yeah.
No. It's a question and answer session later. So I'm very strict. And I really want to give all the speakers
Good.